Hello class, good evening. With a bell ting tong, warm welcome. So welcome to the ninth day of our workshop, which is related to now topic of the leftover topics, that is marker assisted selection, and the last one was transgenic plants and its application. So we will touch these two parts today, and let's see. So let's let people get in, then we start. It's very hard to be sure that there are only 6 members who is watching it, or it's more than that. So during the session, it's quite hard to say how many members are being there. Could you just put thumbs up whosoever is being added so that I could know that these much of members have been added? Just give me a thumbs up that you are part of the workshop today of the series today, day 9th. So I just come in this minute. In the meantime, let others join in. Let me get a glass of water. Good evening, बच्चों. Very good evening. Today I got something more stuff for you related to mass and transgenic plants. Hope you will like it. Uh, from the Americans' point of view, what is why GMO should be allowed? What is not wrong with them? So we will study about them today in more details. About GMO, about mass assisted selected selection. So all these parts will be clear today. So at 8 o'clock or 8:05 we start. So it's 8 already now. So we just wait for 5 minutes more, then we start. Shall we start class then? Marker assisted selection, it's already 8 o'clock. I think we should start. I hope you will all join in between. If not, you can also watch the recorded version later. That's the beauty of YouTube. So, marker assisted breeding for rice improvement. So, in this lecture, we will go through the theory and practice about it and read about various schemes for mass breeding. That is your marker assisted selection. Um, so, let's start with the first part. So what is marker assisted selection? It refers to the use of DNA markers that are tightly linked to the target loci as a substitute or for assess the phenotypic screening. So they are the markers which are tightly to one loci they are targeted and could be found out for further phenotypic screening, whatever the phenotypes that you're looking for, and they are particular belong to that part, and that's how they are so important. And the assumptions are that DNA markers can reliably predict phenotypes in this case. So let's say you have plant 1, plant 2. Plant 1 is a recipient and plant 2 is your donor. So you have a first generation F1, then F2 generation, and after F2 generation you have thousands of plants of inbred of plant 1 and plant 2. So it's time to do the phenotypic selection. So you first is the glasshouse trials will happen, then the field trials will happen. So here you can see bacterial blight screening is happening, and then further phosphorus deficiency plot are being done. So all these things are being taken care afterwards. But in this was the conventional plant breeding. This is what's happening in the conventional way. But in the marker assisted plant breeding, what is happening is this is your susceptible plant and this is your resistance plant. You make a cross, you have F1 plant, you have F2 plant. But in the marker susceptible, when you do the electrophoresis, the marker of that plant looks up after restriction enzyme is come to be above, where the resistance ones comes down. So that's the difference between the resistance plant. And then after the cross, you have large population of consisting of thousands of plants. So here you can see that for the P1 and P2 both markers are coming up. but in this case not. In this case only P1 markers are coming, but in this case we see P2 markers are coming. So that means we will select this part. Same goes uh, here, this one, and this one, and this one. So that's how we select our plant breeding plants, by looking at the markers. So these are the marker assisted selection mass, and we calculated using a quantitative trait. So quantitative trait loci, they are quite near to QTL, and they should be less than 5 centimeter. And method whereby phenotypic selection is based is on the DNA markers. So here the selection is done based on your DNA markers in this case. So advantage of mass is that it's a simpler method compared to phenotypic screening, especially for the laborious screening that we have to do, save a lot of time and resources. Further selection at the seedling stage, important for traits such as grain quality, and can select before transplanting in the rice. Increased reliability, no environmental effects are required, can discriminate between homozygotes, heterozygotes, and select single plants. So these are the various advantages of mass. So what are the potential benefits? So these are shown to you: crossing house and back cross nursery, that how it looks like in general. More accurate and efficient selection of a specific genotype may lead to the accelerated variety development. It has more efficient use of resources, especially field trials. So this is the overview of your marker genotyping. How does it looks like? So in this case, you can see leaf tissue sampling, and then DNA extraction. Then furthermore, PCR is done. Then gel electrophoresis, then marker analysis. In this straight 5 methods from live tissue sampling to marker analysis, your overview of marker genotyping is done. So here leaf samples are being taken, then DNA is extracted, then the PCR machine is done here, then a gen electrophoresis is done, then your marker analysis, that you're looking for polymorphic regions which has differentiated like this one, polymorphic regions. So consider what do you consider while taking the DNA markers in plant breeding? Uh, Reliability, technical methodology, how much is the polymorphism, DNA quality and quantity required, cost matters a lot, available resources are there like equipment and technical expertise is done. So marker must be tightly linked to the target loci. Markers that you're looking for, they should be tightly linked to the target loci. So ideally marker should be less than 5 centimeter from a gene for QTL or a quantitative trait loci. From the gene or from the QTL, they should be, these ideal markers should be less than 5 centimeter. So here, sorry, centimorgan. In this, they are called centimorgan. Uh, this is marker A, and this is the reliability for selection using marker A only. So 1 minus RA give to 95%, QTL 5 centimorgan. Then this is marker A, and then there's a marker B also, and they should be not more than 5 centimorgan, then it's not good. So using a pair of flanking markers can generate improved reliability, but increases time and cost also. So marker must be polymorphic, like in this case P1 and P2, they are like on the same level. So no, not polymorphic, but somehow now they are up, down and up. So they are in this RM296 gene, they are polymorphic. So they are acceptable. So you do that after leaf sampling, wheat seedling tissue samples. It's done in the Australia. Then what you do is in the mortar you make a pastels of them. Then porcelain grinding plates. They are the grinding plates. Afterwards you put your DNA. Then you go for the high throughput DNA extraction with the genome grinder. DNA extraction is done in this way. Then comes the PCR. PCR, as we know, we have discussed it number of times. We add PCR buffer, MgCl2, nucleotide, peptides, tag, primers, DNA template, then polymerase chain reaction with thermocycler. Then it's further go for gel electrophoresis or acrylamide gel. Then we know how we pour the gel, and then we add our samples. Then they run for electrophoresis gel. Then we check them under the special system to check your chemiluminescence or where your band is. Then under the UV light you can check your bands where they're standing. So this is the lab they have shown how people are working in the lab. Then comes the section 2, that is mass breeding schemes. So marker assisted back crossing we will discuss in this. Pyramiding we will discuss. Early generation selection we will discuss in this, and also combined approaches to that. So marker assisted back crossing has several advantage as a conventional back crossing. So you can select the target loci very effectively. Linking drags are not there, it's become less. Accelerated recovery of recurrent parent. So like this is the ground selection, target loci selection, this one. But in the recombinant we select from the up and below. And in the background selection we are selecting from other spots also. So widely used second part is pyramiding is used for multiple disease resistance gene for specific races of pathogens. Uh, Pyramiding is extremely difficult to achieve using conventional methods uh, and considering phenotype of single plant. So pyramiding is extremely difficult to achieve using conventional methods. Consider phenotyping a single plant for multiple forms of seedling resistance, almost impossible, and it's important for the durable resistance against different races. So in this case, in pyramiding, what we are doing is we are combining several genes using two different parents together into single genotype. So there are two different parents, P1 with a gene A and B. Then they have gene A and B in the F1 generation. And F2 we have mass. Then select F2 plants that have gene A and B both. So like this is the breeding plan, and in the genotypes, your P1 is AABB, and your P2 is AA and BB. So the combination is AABB. So you have to select both A and B gene. So for the F2 plants for further. So these are the plants for the same. These are the only selection that you can do is. So this is now called as pyramiding. So early generations mass conducted at the F2 or F3 stage. Plants with the desirable genes QTLs are selected, and alleles can be fixed in the homozygous states. Plant with the undesirable gene combination can be discarded. Advantage is that the later stages of breeding programs, because resources can be used, focus on the few lines. So in this case, like we have the same thing like in the first slides that we saw. You have to select the marker assisted selected plants like this. So for the first QTL, for the quantitative loci, for the first one, let's there will be 75% of elimination of unwanted genotype. So there are some genotypes within the selected markers selected assisted selection, and we don't want 3 out of 4 are removed. In some cases, if there are 2 QTLs, then 94 percentage of elimination is done. Now, out of 16, you remove 15 percentage of it. And this is now your pedigree method has been shown to you. This is P1, P2, you have F1 plant. Then from the F1 you have F2 plant with, and you do the phenotypic screening. So you plants space planted in rows. So you select those plants which are have the phenotypes that you're looking for, and they are grown in the spaces individual plants for selection. So out of them, some are crossed, some are not selected, but some are again grown in the families grown in progeny rows for selection in the pair of four. For out of them again, we're not selecting some. So we take some two plants like here, one plant from here, one plant from here, and one plant from here. And we are not again selecting in the F5 generation. Then in the F6 generation, preliminary yield trials are done. Then we select single plants. So out of all, we select single plants. Further yield trials are done to check. So by this, we do till to F12, multi-location testing, licensing, seed increase, and cultivar release. So we keep checking the things uh, till left to level to have your things that you're looking for. So it's become like a pyramid now, you know. You have big things here, then you're getting reduce to the to your single trait like that. That was pedigree method, and this is a single large scale marker, assisted selection marker, SLS mass, we can say that. Uh, so in this case, only F3 desirable lines are planted in the field, not all, but desirable ones. Then family grows in rows, pedigree selection based on the local needs, what the people need. Then again, it goes to F2 selection. So here, mass assisted selection has been done before we go further. And then the last one, combined approaches. This is including a combination of phenotypic screening and mass approach. Both we are doing it, and this could be quite useful. So this is to increase your genetic gain when some QTLs have been identified from QTL mapping. Level of recombinations between marker and QTLs. In other words, marker is not 100% accurate. To reduce population sizes for traits where marker genotypic is cheaper or easier than the phenotypic screening. So in that case, combined approach helps a lot to us. So marker directed phenotyping, also called as tandem selection. So in this case, what we can see is there is a P1 and there is a P2. So this is recurrent parent and this is the donor parent, and this is susceptible and this is resistance. So F1 generation we have with the resistance gene in the donor parent, then we cross it with the back parents P1. Then this is a cross called as now BC1F1 phenotypes, which has both resistant and susceptible. So they have again in some it will establish, in some not. So phenotypic will be selected further. So in this marker M mass is also done, then phenotype is also selected. So in this way, it save and time reduce for us, and it's used when markers are not 100% accurate or when the phenotypic screening is more expensive compared to marker genotyping. So that's how you do your marker directed phenotyping, also called as tandem selection. So any questions? Sir, please explain the CM. But this was very simple. I didn't went to in details, because that will be too much different part. So I just hit a small portion to it. So this is just a marker A. So this is the uh, so like this is RA, you can say this is RB distance from it, and this is the percentage of marker susceptibility to it, how much it is near. So we calculate this by the formula. So 1 minus 2 RA multiplied by RB. So this is approximately reliability for selection. This is reliable, like how much it will be reliable coming out to be your markers. Then it is 99.5. In this case, if there's only one marker, it's coming out to be about 95 percentage of your marker, and this is just subunit of that we are using in this case. Okay, so any further questions, students? If not, thank you. Well, that was something very small and quite apt, easy to understand topic. And yeah, so let's start the next topic. So these are all very, very small topics, but very important from the transgenic point of view, transgenic research that we are dealing. So this part, so I will be dealing with the some of the success stories in plants. So we are already done with the Agrobacterium tumefaciens. So some diagrams will be there. Maybe I will discuss a little bit. Gene gun we have done quite number of times. So these things might not be discussed in details, but some other aspect will be dealt in this. So something with the GMOs actually that we will be dealing in this part. So this I skip. So food quiz here for all of you. Just look at the plants and find out which are genetically modified organisms. So this is Rio Red grapefruit. This is Minola, Tanglos. This is kind of a vegetable. This is tomato, brinjal, Japanese pear, uh, green beans, some oranges, sorry, papaya. This is peyotes. So out of all of them, which one you think could be GMOs? So answer would be at end of the lecture. So in meantime, have a thought about it. Which could be interspecific species, which could be GMOs, which could not be GMOs, and which could be of plant breeding method, and which could be traditionally there is nothing is happening and something is happening. Just think about it. Later we discuss about it. So plant genetic engineering in history. So this is done been naturally uh, in the genetically engineering plants for centuries. So this is your A, that is your Agrobacterium tumefaciens. This is the genome of it. Here is your TI plasmid with a transfer DNA, weird genes, replication origin, and then it has opine catabolism genes. Then this is a plant cell where it has mitochondria, further chloroplast, and this is the nucleus. So in the step 1 and 2, bacterial cells weakly attaches itself to the plant cells. It then produces cellulose fibrils to anchor to the plant cell. So there's an infection is happening. It just get attached to it. Then in step 3, when the bacterium detects certain compounds produced by the plant in response to bacterial infection, Vir genes started produce various compounds. So Vir genes started to produce various compounds, and then step 4, Vir gene complex cuts that transfer DNA, and from the TI plasmid, from the transfer plasmid, it cuts out, and then in the step 5, in meantime, Vir genes produces compounds that code that transfer DNA to help export to the receptor plant cells. Then in the step 6, now they are being transferred. Vir genes makes the nucleus of the plant receiving the transfer DNA from receptive, and transfer DNA is integrated into the host genome. And then transfer DNA contains the genes that will force the plants to produce special amino acids called opines, which the bacteria can metabolize as its food source. So that's how your opine generated TI plasmid has been transferring your agrobacterium with the help of Ag tumefaciens. So these also producing, they cause gall formation. The TN encodes gene for the production of auxins or ERIA via pathways not normal for plants. So the plants can't regulate its production, only the pathogens, and it's a good code for the production of cytokinins. Together, cell proliferation and gall formations occurs in this case. So we discussed about so far agrobacterium mediated transformation, and then there is also a method called gene gun. Then there is a method called protoplast transformation and fusion that we have discussed in details in earlier lectures. And there is a genome editing, that is a CRISPR-Cas9. So I will touch that part a little bit today. So so this we done. with Agrobacterium transfaciens. This is shown in the tobacco plant sprayed with a chemical substrate luciferin, and they are glowing temporarily for some time. That's the picture done in Science paper 1986. And this is a gene gun done in the Hawaiian rainbow papaya. So inserted a papaya spot virus code protein using high-speed particle bombardment method, and it relies on the pressure. Let's see if this is working. Just wait a second.
So you want to know how a GMO is made? You're not alone. It's one of the top questions about biotechnology. There are different ways plants can be genetically engineered. We'll use the story of the papaya in Hawaii to walk you through one of them. Step 1. Determine whether genetic engineering is the most effective way to solve a plant's problem, such as a disease or damage from pests. For centuries, traditional plant breeding has been used to fight pests, environmental stress, such as drought, as well as to improve taste and nutrition. The reasons to develop GMOs are no different today, but GMOs can be created more quickly and more precisely. Other breeding methods can take decades to produce results and aren't able to be as precise in focusing on one genetic trait or isolating a solution to a specific problem. Since the 1940s, the Hawaiian papaya industry had been battling a problem: the destructive and rapidly spreading papaya ring spot virus, aka PRSV. PRSV deforms the papaya fruit and in young plants destroys the plant's ability to produce fruit at all. PRSV is transmitted by insects that feed on the fruit. Many methods were deployed to try to stop the destructive virus. In the early 1960s, farmers moved papaya production from one Hawaiian island to another, from the island of Oahu to the big island of Hawaii in the Puna district. The virus ultimately reached Puna. Other methods didn't work either, including using insecticides for the insects spreading the virus, giving the plants a mild strain of the virus to create natural inoculation, or destroying infected trees. The virus just continued to spread. 53 million pounds of fresh papaya were being produced annually when PRSV was first discovered in Puna in 1992. By 1998, that amount had been cut in half to 26 million pounds. The crops and livelihood of farmers were being destroyed in front of their eyes. Moreover, for consumers, the availability of fresh papaya and papaya juice for fruit, salads, smoothies, and other products were threatened. A powerful new tool, biotechnology, was now available to fight back. Farmers and researchers turned to genetic engineering, beginning in 1985, for help. Step 2. Identify the gene or genetic material that could solve the problem, like providing resistance to the disease or pest, and study the genetic makeup of the plant that needs the trait. Researchers and farmers had to find a way to make the papaya plant resistant to the deadly virus. PRSV resistance was the genetic trait needed to solve the problem. Researchers knew that if plants contained certain genetic material from the virus within their own DNA, they were protected. If they could insert a copy of this gene from the virus itself into the papaya's genetic makeup, the plants would be protected. Step 3. Copy the trait from a donor organism and implant it into the plant's DNA. Researchers isolated a PRSV gene from the virus itself. Then they needed a way to get the genetic material into the papaya plant's own DNA. Researchers deployed a commonly used technique in biotechnology called transformation, a process by which a copy of genetic material from a specific trait is inserted into cells. There are many ways to transform a cell. In this case, the researchers used a high-speed particle bombardment method. Through a high-pressure insertion device, the gene from the virus was inserted into the DNA of the papaya seed. The genetic material made the plant resistant to the ring spot virus, just as a flu shot makes people resistant to the flu. The researchers were able to successfully transform 17 plants. These plants contained this gene from the virus and produced a protein from the virus that helped the plants become resistant to the PRSV infection. Step 4. Plant the new seed and test it. The final step was to plant the seeds and carefully test the resulting plants. One line of plants demonstrated consistent resistance to the virus. After several years of testing, it was determined that this line performed well on the farms, was safe for people to eat, and was of commercial quality. Success was achieved. One variety of transgenic papaya, the rainbow, yielded about 125,000 pounds of marketable fruit per acre per year, 25 times the amount of fruit produced per acre by a non-GM variety infected by PRSV. These tests took about 7 years, including food safety and environmental review by the FDA, USDA, and EPA. Once regulatory approval was gained, the Hawaiian farmers chose to start using the new papaya seeds. The result? The harvesting of the new PRSV-resistant papaya began in 1998, and by 2001, the industry had rebounded, producing 46 million pounds of fresh papaya, free of PRSV. This kept the vibrant papaya industry alive in Hawaii, and ensured consumers had access to high-quality papaya and papaya juice at a reasonable cost. Genetic engineering allowed the introduction of PRSV resistance without altering other traits of the papaya or changing the plants in other ways. The plants were exactly the same in nutrition and safety. As of 2010, 70% of Hawaii's papaya industry is GM papaya. GM papaya has been grown and consumed in the U.S. for more than a decade. This is just one GMO story. We hope to share more with you. Please share your comments.
So, a very interesting story about Yersara virus, how they produced rainbow papaya, and which being produced in the Pune area of uh, in Hawaii. So that's how things are done with the help of gene gun. Very interesting story that GMOs are not so bad and they are being widely used. Then comes the gene silencing technology, that is your siRNA, RNA targeted, where you knock down one gene and then you see the specific gene that you're not required, you can find the results out of it. There's a protoplast fusion mechanism in which protoplasts of distinct species are fused together to produce a new plants and having a characteristics of both plants. So like somatic hybrids of solanum and mechocanum bitter, and you cross them together. And then also you cross tubularism with autofuse with the mechocanum plants. So you produce a source of the late blight resistance of plant branding by Smith et al. So you can produce a plant disease resistance between crossing of different species of protoplast also. Then comes the last technique using CRISPR, that is your clustered regular interspaced short palindromic repeats. So what is happening is in this case, uh, so there's a guided RNA, gRNA, is attached to Cas9 protein, and then a bacterial enzymes that will cut the DNA sequence at the desired site in the genome. First, the guided RNA, which is attached with the Cas9 protein, it will cut the desired DNA from its DNA genome where it has to be cut. Once the genome is broken, the guided RNA Cas9 disappears, and the cell will try to repair the cut, and which can be disable and knock out the particular gene. So scientists can insert of then a new segment of DNA into that cut region, essentially pasting a gene into the desired location and changing the genome. So CRISPR system was first discovered in bacteria and function as a defense against foreign DNA, either viral or plasmid, and is used for the gene knockout, repression, or activation. And it has some limitations, but because of the precision and simplicity, it is a genetic tool of the moment at the moment. So to say about it a bit more, let's have a little bit of virtual lab of CRISPR-Cas9, how basically it works. Let me see if I have any video. No, I don't have. So let it load. It will take some time, I guess. Not much. So as we can see, mainly the gRNA, your gRNA has a Cas9 attached to it. It goes to desired place where it has to cut. Then after cutting, then you add your desired GNA to be added, or you want to keep it cut, then you remove that part. You can do addition, deletion, or mutation, anything that you're requiring for. And it's a quite easy method to do so in this age, and it has been widely used in the at the IITs also. At the IIT Bombay and IIT Kharagpur, they are being widely used at these techniques, CRISPR-Cas9, even in the ISC, Indian Institute of Sciences in the Bangalore. So everywhere they are quite extremely usable. So almost done. Let's wait for 1 more minute. Okay, you're almost done. Oh, nice. So students, please go read through this. So this is your CRISPR-Cas9 protein. This is your cellular DNA, and this is your guided RNA. Okay, here's your guided RNA, Cas9 cellular DNA. So we have induced into the system. So Cas9 binds to a 3 nucleotide sequence motif called PAM, and which is abundant in the through the genome. So it will find a sequence called PAM, PAM, Cas9. So here is a PAM came. It will attach to it and unwind your DNA by attaching to it. So once bind PM motif is attached, Cas9 protein will unwind the DNA double RNA helix and then matches a sequence of about 20 nucleotides within the guided RNA. And the DNA and matching RNA will bind through the complementary base pairing. So now after unwinding Cas9, your guided RNA comes there to wind up with the target RNA. So from there you can see it belongs this gRNA, it's got attached to it, and then it goes further. Then DNA RNA pairing triggers Cas9 to change its three-dimensional structures and activates its nuclease activity. So Cas9 will cleave both DNA strands at site upstream of PAM. So they cut it like that. So cell contains the enzyme repair DNA breaks. The repair process is naturally error prone and will lead to mutations that may inactivate the gene, cleaving DNA at the precise location and a many application of the CRISPR-Cas DNA technology. So DNA breaks, so new DNA will come to repair system and will join it. So here your mutation is done, your target DNA is there added in front of a PAM, and this is your repair DNA sequence also. So all done. So that's it. That's how your CRISPR-Cas9 works in 3D level. So I will share this with you all. How GMOs are made from the papaya story. Okay. So let's continue. So now there is a website called GMO Answers, created by the scientists and independent experts who answer the consumer questions and which are not paid to answer these questions. Experts then they donate their names to these questions from the expertise of the website, and they're passionate about helping the public understand GMOs, how GMOs growing. So website name is gmoanswers.com. So it has been supported by many companies and many associations, such as Minnesota Crop Production Regulators, National Association of Wheat Growers, American Seed Trade Associations. So all are being related to GMOs crops. Sorry. Uh, so in the history of genetic modifications, so around 10,000 years ago, human begins domestication of using selective breeding. And in 1700s, they started to do crossbreeding. Then in 40s and 50s, breeders and researchers to introduce genetic variations into the pool of plants. And in 80s, started to have more precise and controllable methods of genetic engineering. Then in 1990s, first GMO was introduced in the marketplace. So these are the various examples that has been produced as GMOs, such as watermelon, corn has been produced like that, banana, such kind of bananas were produced, eggplant in the circular shape with different color, carrots like this, and cabbage, kale, broccoli. They are all examples of your further GMOs. So this, what is this chart? It's comprised of the modern methods of seed improvement and how do we create new plant improvement of varieties. So let's I just zoom in. Uh, so I hope you can read it now. Um, yeah. So here is like selective breeding. Here is interspecies crosses, mutagenesis, and this is the transgenes of GMOs. So in this, what is happening in the selective breeding? We are combining the trait from the similar or dissimilar plants by crossing into one genetic background with improved traits. In the case of interspecies, we are crossing the two species genetic exchange, but not cross naturally. Then the mutagenesis, which is started in the 1930s to today, so we use chemical radiations change to occasionally induce favorable traits. In the 1990s to now, this GMOs now we are working, we add specific characterized gene to new seed to gene specific trait. So some examples are almost whatever we eating, they are because of the selective breeding. Interspecies is like glucose, tangleose, some apple, rice, and wheat. So many plants, fruits including pears, apple, rice, jams in mutagenesis. And transgenic GMOs are like cotton, papaya, potatoes, soybeans, apple are approved coming to the market soon. So, do breeding improved? Yes, in all these cases. How many genes are affected? So there are many genes, around 10,000 to 300,000. 10,000 to 300,000. Random, unknown. We don't know how many mutagenesis are being affected. But in this gene GMOs, we are know that we are particularly affecting these genes and we are not affecting any other genes. So in this case, only one to two, three genes are affected. And as we know which genes in the seeds are affected, in this, in the our three case, we are not aware of it. But in the fourth case, yes, we are aware that yes, we are affecting these genes. So research and development time, so they take 5 to 30 years, 5 to 30 years. This take mutagenesis 5 plus years, and this also take 5 to 10 years, a bit less time. So tested by regular agencies to ensure safety for people, animal and environment. In the fourth case, yes, GMOs are being taken care. Can seeds be patented? Yes, they can. So can non-GMOs approved for organic farming? No, in the GMOs not, but other yes. And are people asking for labeling? In the fourth case, yes. In the one third case, no. So this is how you can check that you can see that a lot of advantages of GMOs. People are just afraid of them for without any reason. There's nothing to be worried about actually. So the type 5 misconceptions that we see across social medias are that if it is extra large, seedless, look weird taste, but feels squishy, it must be GMO. So whatever things that are not normal, so it is GMO, we should neglect it. GMOs are not safe, they are tested by companies making them. So they say that's company thing. There is animal DNA in GMO, some says. They say they insect pesticides into them. GMO companies force farmers to grow their crops or sue farmers if GMO seeds are blown into their feeds. So these are the quite in the social media. Uh, so it's like that. So this is like normal way of growing of a strawberry, but people says they are tweeting it like GMO strawberry, still gonna eat it. So it's not, they are naturally grown. Sometimes they got hybrid and they grow like that. So who is responsible for this? A cartoon for the penguins. Everyone is saying that everyone says to each other for GMO safety testing, but all of them they're working together for it. So these are the various misconceptions. Uh, so how can we ensure that GMOs are safe for use and consumptions? GMO crops are studied extensively to make sure that they are safe for people, animals, and environment. And GMOs can take average of a $136 million and 13 years to bring to market because of research, testing, and regulatory approvals conducted by government agencies in the United States around the world. And these are some National Academy science reports of various fields. So I will not go through them. So you can produce currently in the USA, there are maize with herbicide tolerance and insect resistance. There's following plants are being produced: sugar beet, alfalfa, papaya, disease virus resistance, potato, and apple. So genetically modified crops. So out of this, so we have this question before. Now there's the answer. So these ones, Rio red grapefruit, they are derived by mutagenesis, and these are no interspecies cross. So they are sorry, they are not GMOs, but they are interspecies cross, that is between grapefruit and tangerine. So these are the GMOs. So they are derived from mutagenesis, Japan pear. So they could be maybe. This is not no, no. This is yes, and this is no. This is also interspecies between apricot and plum. Together they are grown together. So that's it from this perspective. Any questions, students? This I shared, right? This is also I shared. Any questions so far? So I will touch the last topic of today. That is uh, day 9. Transgenetic. So what is transgene? It is a gene or genetic material that has been transferred naturally or by any number of genetic engineering technique from one organism to another. The process of introducing an exogenous gene called transgene into living organism so that the organism will exhibit a new property and transmit the property to its offspring. So transgenic plants, the plants which express the characteristics coded by transgene are called transgenic plants. So traditional breeding, it is done by selective breeding, cross pollinations, having various disadvantages like long processes, lots of manpower, limited possibility of improved rates. Transgenic technology versus traditional. Transgenic technology is a gene from the any source like animal, bacteria, or a virus, whereas a traditional breeding moves or genes only between the members of particular genus of plants. So why transgenic plants? Because we have crop improvement, disease resistance, stress tolerance, improved performance, value-added traits. Basic studies are done like gene expression, reverse genetics, biochemistry, metabolism. So transgenic plants whose genome is altered by adding one or more transgenes are known as transgenic plants. The phenomena of introduction of exogenous DNA into the genome to create maintain a stable heritable character. So first in 1982, antibiotic resistant tobacco plant was produced. Then genetic engineering Colymovirus vector has been used. Then in 1994, first flavor saver tomato was produced in the world. So we understood how we created transgenic plants with a first selected target genome, then a vector, then foreign DNA to increase the level of gene expression, method to deliver it, methodology to transform the cells, then tissue cultures like Agrobacterium tumefaciens. It has been approved by addition of one or more following genes. Endotoxin genes could be added like in the Bt cotton, protease inhibitors, alpha amylase inhibitors, lactin enzymes, pyrimidine genes. So all are the example of that. So we already know vector mediated and direct with the gene gun. So I will not go into these details. We have done them quite number of time now. Gene gun method. So there's applications of transgenesis, that is edible gun, we can edible vaccine, sorry. The genes encoding antigens of bacterial and viral pathogens can be expressed in plants in which they retain native immunogenic properties. So the edible vaccines, transgenic plants are used as a vaccine production system. So recently people have started also to produce vaccine instead of bacteria or viruses, bacteria or other cells. They try to produce in plants also, and they could be act as your edible vaccine. So you can just, instead of getting a shot, you just have to eat the plant to have it. So vaccines could be prophylactic and therapeutic. Prophylactic are the one which are designed to prevent the disease from occurring. For example, there is a rheumatic fever to prevent the subsequent development of Sydenham chorea. Whereas a therapeutic is the one once the disease has happened, then to induce the antiviral immunity to alter the course of disease, to remove that virus out of our body. So you can do it in two ways. In one case, entire structural gene is inserted into plant transformation between 5' end and 3' end, and this will allow the transcription, accumulation, and coding in the sequence. In the second phase, epitope of the antigen is added to the DNA fragment, and coding these can be used to construct your fusion or code genes of the plants like TMB or CMB. So here, like, you have one is plasmid vector, one is the infection of the plants with plant virus. So structural DNA loaded in the plasmid vector is added to the plasmid vector, then gene transformation is done, and then transgenic plant is there. On the other side, you have a DNA encoding epitope inserted into viral genome, then infection of plant with the plant virus, an infected plant is there. Then extraction of your antigen stop production, then you have edible vaccine, and it could go for preclinical studies. Uh, so this is how your vaccination works. Actually, initial response, what happen is you have your M cells. So your antigen from vaccines, so they are coming. So in first case, your M cells pass the antigen to the macrophages and B cells. So it will give it to the macrophages and B cells, these antigens. Then macrophages will display piece of antigens to the helper T cells also. And then the T cells stimulate B cells, seek out the antigens at the distant sites. Then fourth, activated B cells will make release of antibodies to able to neutralize the antigen. They will produce the antibody to release the antigen, to make neutralize the antigen. So when a disease agent appears, so whenever the virus came, arriving virus is there, so what it will do, first of all, cytotoxic cells. So memory cells will produce cytotoxic cells to attack the infected cells. And then memory helper T cells swiftly stimulate antibody secretion. and then they will antibodies quickly neutralize the invaders. So the antibodies will go and they will attach to the virus and will not let enter inside the body. So that is how your vaccination works actually in your body. This is one case study, go through it. And these are some clinical trials, current studies. In 2008, hepatitis B was produced in the potato-based vaccine against hepatitis B and reported for encouraging results. So amount of hepatitis and antibody needed for one dose could be achieved in single potato when cloned in CMV, plasmid hepatitis and antigen subtypes showed higher expression in roots as compared to the leaf tissue of the transgenic potato. So they could also, your transgenic product could act as a bioreactor in which organisms could cultivate in the desired product. So, oops. Transgenic plants have significant potentials in the bioproduction of human therapeutic agents due to ease of genetic manipulation, lack of potential contaminations with human pathogens. So there are types of plant bioreactor, that is plant suspension culture, chloroplast bioreactor, hairy root system, seed-based bioreactor. So first is the select design gene high level expression. Then you have plant transformation, then regeneration of the selection or regeneration of the cells, then select the transgenic plants, you growth of a plant in the field, harvesting of plant material, purification of desired products, then biosafety and functionality test. This is how we do the transgenic plants as a bioreactor. You can also use them as a bioplastic, such as polyhydroxyl alkanates (PHAs) or PHBs formation in plastids or PHA formation cytoplasm. And also you can do with the help of fish, Antarctica fish called Arctic flounder fish, from which we get the glycoprotein, it helps to lower the freezing point of strawberries and which has a problem freezing at the freezing conditions. So what they do, they put this antifreeze protein into the strawberries to increase the frost resistance to the cold things. So this is how they call Kirk EMSH. And also transgenic plants are grown against the biotic and abiotic stresses. So we have discussed them in a couple of times in our previous lectures. So like wheat carrying GM-DREB gene from soybean under ubiquitin promoter should show drought tolerance. Uh, and one way is inserting genes for compatible osmolytes such as sugar and amino acids. Or you can constitute the overexpression of the late embryonic proteins which provide the dehydrations. So these are the various advantages, various disadvantages of transgenic plants. So in conclusion, transgenic plants have potential to solve many of the world hunger and malnutrition problems, help to protect and preserve the environment by increasing the yield and reducing reliance upon chemical pesticides and herbicides. So students, thank you very much. Last thing I want to say from today's plant biotechnology chapter is over, and we mature with mistakes, not years. So we do a lot of things that we should not do, and we learn out of it, and then from there we are getting better and better. That's the end note of our thing. So I will add these lectures to it today. Day 9. So students, how's your review is going on? Because without review, your internship letter you will not get. So you will send your topic and number of students who have done it. Please share that in the group. Only then you will get your certies and internship letter. So whosoever has registered for the review, so please take care of that. Otherwise, there will be delay in your getting your certificates then. Other persons, I'm getting your nice things, so your nice assignments, so they're pretty good. So please sharing your assignments also with me so that I can go through them. And for tomorrow I will teach you, I have decided that is it okay if I want to teach you bioinformatics and protein isolation and purification? Just tell me yes, or if somebody wants to have add something more, please let me know. because I can only do add these topics for tomorrow, because they were also, because protein isolation and purification is common to animals and humans. So I decided with all the techniques, proteins are the essential part of our studies. So I thought protein isolation and purification will be quite essential to teach you before you all leave. So that will be taught to you, and also bioinformatics, little bit of bioinformatics also. So tomorrow will be your last class, and it was good time with you all to teach you a lot of interesting things about plant biotechnology, and hope to keep you in touch with you all of you. And let's see, little information about proteomics. Okay, I will see, I can touch that topic also, don't worry, I will touch that. Some introductory class for proteomics then. Okay, so today's lectures are done, are all shared. Yes. So, wish you all good night. Stay safe. I will again repeat, don't go outside much. Stay at home. Be aware of corona. Wash your hands. Have a mask on your mouth. In India, it's really horrible at the moment. So, please take care of that. Okay, then. Good night. See you tomorrow.